Thank you so much, Robin, and thank you to Save the Great South Bay for the invitation. Pleasure to be here. Nice to see a lot of familiar faces. Yeah, so let's get started. I'll start by just acknowledging my lab group at Shinnecock Bay University. These are the people, these fine young men and women are the people who are the boots on the ground and the fins in the water who really make everything happen here. So want to definitely give them all the credit. And from there, just go on to talk about Long Island as a watershed. I think we're all familiar with this concept. And it's really a grounding concept for us here living on an island, and that our drinking water source comes from below our feet. And therefore, what we do on land has a direct impact on the water we drink every day. And what we don't extract to drink, of course, discharges into our surface waters. So that groundwater is doubly important for us all here on Long Island. And as Long Island has grown, the characteristics of that groundwater has changed. Just showing that we've gone from a rural setting to a very heavily suburban setting across Suffolk County. And with that, we've had the rise in levels of nitrogen in our groundwater, and to levels that are now on average 3.8 milligrams per liter. And we know the research has been done for all of Suffolk County on average. The largest source of that nitrogen in our groundwater is from on-site wastewater, although fertilizer is the second most important source. And I'll point out also it's really important to not presuppose. There's the Suffolk County Subwatershed Study looked at more than 200 subwatersheds. And so there are some where fertilizer is more important. But by and large, wastewater is the biggest source. Now, on the fact of the levels of nitrogen in our groundwater, I want to just put that in some national perspective. This is a paper from Duke University that came out a few years ago. And if you look at the graph there on the y-axis, that's millions of people, so 200 million people in the United States, 2/3 of the country nearly, have groundwater with less than 1 milligram per liter of nitrate in it. So you can see we're sort of in the tail end at high levels, and there's a lot of variability in that. If you look at this map of Suffolk County, you can see areas, for example, the Pine Barrens that are very, very low, but you also see areas that are far above that 3.8 milligram per liter threshold. And so, 3.8 milligrams per liter, well, that's below the EPA standard for drinking water. And that standard was created for something called blue baby syndrome, and the fact that if you have high levels of nitrate, particularly in infants, that can compete with oxygen uptake. But there's emerging epidemiological research that nitrate in groundwater at high levels might be problematic. This is just a graph showing that this is a hot research topic. This is showing the number of papers published and the number of citations on the on the topic and scientific research of nitrate in drinking water and cancers. And so specifically, I'll say the World Health Organization actually considers nitrate probably carcinogenic. And there's a whole fleet of research studies that have come out recently showing links between high nitrate in drinking water and different types of cancers, usually gastrointestinal, but not always. And I'm showing the list here, and I'm highlighting, just in 2024 alone, a whole suite of papers. And you can see this is, again, just going back here, this is really emerging. Like, no one thought about this, but in the last few years, particularly, less than the last 5 or 6 years in particular, it's really an exploding research topic, one that hasn't been on the radar of many people. It's also connections with adverse birth outcomes and high levels of nitrate in drinking water. And I'm just going to highlight this one paper that came out recently out of Denmark. Denmark's a small country, it has 6 million people. But essentially, this paper in particular showed that if there were lower levels if the standard for nitrate was lower, that not only would it save lives, it would save a lot of money. And again, we're a country of over 300 million people. This is 6 million people. Again, going to the same researchers out of Duke, pointed to almost 13,000 cases of cancer per year in the U.S. that could be attributable to high nitrate in drinking water. It could save, if we had lower standards, $1.5 billion a year in healthcare costs. So I'm not an epidemiologist. I work with several at Stony Brook University at the hospital who also consider this, who actually turned me on to this research. But I think the upshot of all of this is that we want our nitrate levels in our drinking water as low as possible. But because beyond that, there are a whole host of water quality impairments associated with high nitrate getting into, and high nitrogen getting into surface waters. And here's a map that's really hot off the presses. It says draft, but it's actually the final version. This map shows the occurrence of, it just finished, harmful algal blooms that occurred essentially between April and September of 2025. It also shows areas of low oxygen. And what you can see is something we've seen all along. You can see these things are spread from the tip of Montauk all the way into the city. And you know, in 2024, we've had more blue-green algal blooms in our freshwater bodies than ever before. So 30 locations, that's a new record. We also had, and I'll describe it, a record-setting paralytic shellfish poison event, and more than 34 low oxygen or dead zones found across Long Island, just, again, this summer alone. And on the harmful algal bloom front, you know, the ones that we're detecting are both the ones that can make toxins and also some that we call ecosystem disruptive, that don't make toxins but are harmful to the ecosystem. And there's a plethora of research showing that high levels of nitrogen stimulate the growth of many of these harmful algal bloom species. And in other cases, high nitrogen allows them to make more of their nitrogen-rich toxins, specifically things like microcystin and saxitoxin. So I'm going to start talking about the freshwater situation and blue-green algal blooms. Here's Lake Agawam, and you can see that's not the green of a golf course, but rather that is the lake itself. And you know, these are serious events. The most common blue-green algae or cyanobacterial bloom that we see in our freshwaters are caused by something called microcystis, and it makes the toxin microcystin. This is a gastrointestinal toxin. It's been linked to cancers before. And this is just one example. There are many other species and types that we see and other toxins also, but this is the most common. And it's potent. So for example, here's an event that happened in 2020 where there's the death of hundreds of endangered elephants on the plains of Africa. And you know, the final analysis was that this was because ephemeral or seasonal freshwater bodies and ponds, as they were getting smaller and smaller, got to a certain volume at which the nutrient levels were very high and there was an explosion of these algal blooms, and that was the cause. And so you know this is a potent toxin if it can take down you know, the largest land mammals on the planet. Closer to home, we've seen incidences of dogs getting sick or poisoned by these events. There's usually at least one per year of a dog at least getting sick on Long Island. If you follow the national news and you looked across the country this summer, there were dog deaths. We had a dog die in East Hampton, and this is not surprising given, you know, CDC has done studies essentially showing there's been hundreds of these cases over the years. And then emerging research shows that some of these toxins, specifically microcystin, can be aerosolized, right? So even if you say, well, I'm not going to go in the water, if you're nearby and the conditions aren't correct, you could actually be inhaling the microcystin. This is ongoing research. This was recently published in Environmental Science and Technology, but there's people at Stony Brook University who are keying in on this as well. I'll just emphasize, I'm in the School of Marine and Atmospheric Sciences, so we have some atmospheric scientists who are looking really carefully at this, and it's definitely happening. So it's really a matter of, you know, what's the dose in that case. The DEC does a great job of tracking these events all over the state. And in 2024, they haven't released all the 2025 data, but in 2024, 56 of the 62 counties in New York had these events, but no county had more than Suffolk County at more than 20, and this year we were at 30. So, and that's a consistent trend. That happens nearly every year. An emerging yet another issue with regards to these toxins is that it looks like they're not staying just in the lakes. If you have a freshwater body that's connected to an estuary, This was actually really groundbreaking and frankly kind of shocking research that came out 15 years ago, but it really opened up a whole new avenue of research. There was the die-off of sea otters in Monterey Bay. And no one knew what was happening. But upon doing autopsies, I mean, I don't know how many people would recognize what a healthy liver looks like. But this is, here is a bloodied liver, right? Remember, microcystin is a gastrointestinal toxin. And so through the research, they were able to actually link that very intense blue-green algae blooms within the watershed were flushing into the estuary, and the toxins were accumulating in the shellfish. They were being consumed by the sea otters. And so it's the idea that these toxins don't, even though they're fresh water, and we may say, oh, who cares about fresh water if we're interested in the bay, they can be transported. And there's, I don't have time to go into the reasons why, but because of the way the cells form and the toxin being stable, they can potentially make their way into coastal waters. And we've actually looked at this in some areas across New York, downstate New York in particular. We have a couple of publications on this that came out earlier this year. I won't go into the details, but I'll just simply say that it does happen. Right? So this is yet another concern with regards to these blue-green algae blooms. And these papers, I'm going to point to a bunch of papers through my talk. They're all freely available. So if you're interested in these, you can just go and download them and read the details. But we do know that both nitrogen and phosphorus are important for these blooms. And in the nitrogen front, I kind of alluded to this before, if you look at that toxin that we're worried about, it's a very nitrogen-rich compound. And again, so more nitrogen allows the cell to make more of this toxin, and it works in reverse. And we've shown this. We've done transcriptomic studies looking at gene expression. You can turn the toxin synthesis genes off just by starving this thing of nitrogen. Okay, and on the marine front, the harmful algal bloom that we're probably most worried about are Alexandrian blooms. Alexandrian makes saxitoxin, which is a neurotoxin. That neurotoxin is 1,000 times more potent than cyanide. And if it can get into shellfish, it can lead to something called paralytic shellfish poisoning, which can be a very serious outcome. I'll leave it at that. You can see the headlines. This organism is widespread across Long Island waters. Maybe difficult to find in open waters, but when you go into inland waters, it's present and often at high levels. It's seasonal, but it can reach very high levels. Now, up until this year, what you can see here is that we had 8 different locations across Long Island that have been closed to shellfishing due to high levels of saxitoxin. And many of these locations, it happens many years in succession. Although there's variability, some years there are no events. But in 2025, we had some particularly bad closures. For example, the entire western half of Shinnecock Bay was closed to shellfishing due to high levels of saxitoxin in the water and detected in shellfish. This organism has been present for decades there. 2025 was the second highest level we've ever seen with regards to cell densities, and this is the largest shellfish bed closure that's occurred there in 15 years. There was also an area in Moriches Bay, so the second South Shore estuary site, and the far eastern extent of Moriches Bay that also had an event and led to a closure. And then in the Peconic estuary, there's a particular event that was pretty troubling in that, first we had a closure of 2 locations, places called Jockey Creek and Town Creek. That was then expanded to an adjacent creek called Goose Creek. These began in April. They didn't reopen until August, right? And to me, this was completely shocking because typically these closures, well, actually, when I show you some more data, if I didn't know more about it, it'd be completely shocking. But when I show you some more, it will make more sense. Typically, these closures, these events are seasonal. So in happy news, they happen in the spring. The waters warm up, everything's gone, and everything is fine. You can see in this particular event, this went on for many months, almost 4 months of a closure. If you can see this image, you can see, particularly this is within Jockey Creek, the one particular area, which is the epicenter where these blooms were happening. They were just spreading into these other systems. You can see the discoloration of the water. We went out there and we found actually what 11 million cells per liter, did thorough checks. There's never been this high level ever recorded. I thought in New York, I knew in New York, but actually in checking in the United States. I checked against everything in the Northeast and on the West Coast. Might have missed something, but I guess it's not that important. We've never seen a level this high before, and you can see the discoloration of water. You can see what the sample looked like under the microscope. And just on the saxitoxin front, 80 micrograms per liter is the closure limit set by the FDA. Up until this year, the highest level we ever saw was 1,100 micrograms per liter, 1.1 milligrams per liter. And in this bloom, we exceeded that almost by double at 1,800 micrograms per 100 grams of shellfish tissue. So a very intense event, very toxic event that was very long-lasting. And we've actually done research in this exact system. So here's experiments that we did just 2 years prior, where we took the water, we did nothing, or we added nitrogen, phosphorus, or just phosphorus. And you can see in just 48 hours, we could triple the number of cells that we had in our experiment with the addition of nitrogen. So, clearly a strong link there, consistent with prior research that's been published. This is papers that are 10 and 15 years old, where we've shown high nitrogen gives you more cells and actually more saxitoxin. Saxitoxin is the other nitrogen-rich biotoxin made by algae. An area that used to have a lot of these events was in Northport, specifically emanating in Northport Harbor, but then in some years covering all of Northport Harbor, Northport Bay, Huntington Harbor. In fact, the largest shellfish bed closures we've ever had in New York State. In 2013, in Northport, they upgraded the sewage treatment plant there to significantly reduce the amount of nitrogen being discharged from that sewage treatment plant. And so before that upgrade, you can see this graph is showing you the acreage of this whole system that was closed to shellfishing each year. And in the year after the upgrade of the sewage treatment plant, there have just been 2 closures, and they've been small. And we can add 2025 to this list. So, and there's a few caveats here. The one is that there are other strong nitrogen sources here, but this plant in particular was discharging right into the back of the system, right where we know actually was the epicenter where the blooms were and are. So it's a really bad location. Some of this also could be related to changes in monitoring by DEC, but the cell densities that we measure also track with the closures. We're seeing lower cell densities now as well. A final thing I'll talk about with regards to high nitrogen and adverse outcomes is low levels of oxygen. It's well known, more nitrogen gives you more intense algal blooms. That's more organic matter that will then be broken down by bacteria. They consume oxygen, and that can lead to low oxygen levels. And the DEC has, via the Clean Water Act, and that being promulgated to the states, has very stringent and very clear quantitative standards on oxygen levels. If you look at actually water quality parameters, there's very few things that are regulated with regards to water quality. Like that, there's not a nitrogen level that you can't exceed in the water, or a phosphorus level, or an algae level, but there is an oxygen level. And so what you can see is there's very clear standards, and we never want to get less than 3 milligrams per liter. In fact, when I showed my map of the low oxygen zones on Long Island earlier, that's the standard we use, the lowest one. In 2024, you can see there were very few sites that actually measured up to that level. And the question is, does it matter? Right? We have, I told you this year as well, we had 30 some odd sites with low oxygen. Perhaps this is of no consequence. We just published a study, again, you can download it yourself and read it yourself, to look at, does this matter? This is by my graduate student, Jeff Kramer, Sternberg University, where we worked in a location that we knew had low oxygen at night, and we specifically set up a sea table where we're sending the water through a maze with lots and lots of aeration before then delivering it into chambers that had bivalves in them. And then we also just gave the bivalves the raw water. So the water we fixed, and what are the ambient conditions, does it matter? So this is what oxygen looks like in our coastal waters. Just an example. This is actually fairly mild. this particular location is the Southampton Marine Station. And you see there's day-night variability, and that happens all year long. It's very acute in summer, however, where you get high levels during the day due to photosynthesis, low levels at night due to respiration. And so if you look at the data here, yeah, we're having low oxygen, but it's only for a few hours, and it's only at night, and, does that matter? And again, you can see the blue is where we fixed it, where we never get below that standard. So is the standard, what is the standard telling us, and does it matter that we're going below it? And the answer is, yeah, it does matter, and that we're significantly slowing the growth of bivalves when they're exposed to this low oxygen. You can see here we can come up and nearly double the growth rates of, in this case, oysters when they're receiving that fully oxygenated water all the time, right? And again, remember the control here is mild. It's just they're getting sort of low at night. That tells you that standard is actually pretty good. And the fact that we're breaching that standard in many locations is consequential. And this plot on the right is just showing you the improvement of growth rate when we gave the bivalves the higher levels of oxygen. Jeff then went and looked at all the oxygen levels across sites all across Long Island we had records for, and was able to model what is the reduction in growth rate due to the low oxygen conditions. And essentially what you can see is that, the ones that we were experiencing, where it's maybe a 50% reduction or a 30%, was nothing compared to some other locations that we know have chronically low levels of oxygen, right? That might be getting down to as low as 0 at night, and it's extended even longer. So very clearly, this has an impact on bivalves, and it also has an impact on restoration. This is just showing you the survival rate of bivalves depending on their size and age. So when we put out adult clams, we get almost full survival, and I'll talk about this later, where we have clams that are now decades old and they're still doing their thing. But the survival rate of the juveniles is wholly size dependent. If you're putting out seed clams of less than 10 millimeters, they're not going to survive. Right? And you can see even when they get up to larger sizes, here up to 20 millimeters, yeah, you'll get some survival, but it's really, really low. That's why when we've done restoration at Stony Brook, we're using adult clams, not juveniles. But this is the consequence of oxygen. Right? If we have slowed growth rates, the clams need to get to this size. If their growth is slowed, that means they're going to be more subject to mortality. Okay. I'm going to change gears here just a little bit to talk a little bit about changes in climate, and specifically the Anthropocene, the idea that we're in an era where human activities are a dominant force on climate and the environment, because when we think about our coastal ecosystems, we need to keep in mind how climate change issues might be affecting them. So just as a review, I'm sure we all know that 2024 was the warmest year ever on planet Earth. It was the year we exceeded 1.5 centigrade of warming, which is the level that many scientists said we should not breach. And it was hard to find a part of planet Earth that wasn't significantly above average in 2024. And even in 2025, actually January of this year was the warmest month ever. Now we're transitioning, thankfully, into a La Nina season that should temper things globally. But frankly, it's not the global averages that matter, it's what's happening locally. But, on that front, if we look at our coastal waters, a paper published by my student Stephen Tomasetti a few years ago, essentially showing that if you look at our summer water temperatures, they're not only are they much higher, but they've increased at threefold the global average. And that's just since 2002. So there's been a lot of warming in a short period of time. And beyond the temperatures itself, and I could, enroll at Stony Brook, I can give you a whole lecture about consequences for marine life as water temperatures change. But one of the consequences we know beyond the temperature itself and how that affects organisms is that simply leads to more moisture in the atmosphere, which can lead to more rain. And we know for a fact, since the 20th century, we've had a 71% increase, not in rainfall, but in all-at-once rainfall. These very, very intense events where we get many inches of rain falling all at once. And just to highlight, for example, I don't know who remembers, but in last year we had 10 inches of rain fall, and you can see that was like there was an epicenter there right at Stony Brook. Stony Brook actually couldn't open its dorms. Several dorms couldn't be opened on time because they actually were flooded because of 10 inches of rain all at once. And people may know the Stony Brook pond doesn't exist anymore because it was washed out by this event. Blydenburg Park or Blydenburg Lake doesn't exist anymore. It was washed out by this event. And it was called a 1 in 1,000 year event. Although when I saw that, I said, 1 in 1,000 year, 10 inches of rain, is that right? And I went back, I only had to go back 3 years, and we had Hurricane Ida, where it dropped also about 10 inches of rain or more. And of course, this is the trivia question. Who remembers the event in Islip in 2014? Anybody remember that event? Okay, well that is the record in New York State. 14 inches of rain fell in one overnight, literally. So there again, it was called the 1 in 1,000 year event, but you can look back 2014. That's 3 events in 10 years. And that's actually consistent with this paper that came out recently, essentially just showing that these events that used to be 1 in 100 or 1 in 1,000 are now happening a lot more frequently, and you can see where the epicenter of it all is. We're in the bullseye of it. And so these 100-year or 1,000-year storms are now happening not just decadally, but every 3 years or so. And that has consequences for nitrogen loading, right? Where there's more fresh water going from land to sea. This is my colleague Anna Michalak from Stanford University, published this paper in Science just a few years ago. Firstly identifying, the Northeast, we're sort of at the epicenter of it all, but that going forward, the changes of precipitation are going to lead to actually a significant increase in nitrogen loading as well, meaning that the do-nothing path leads to more nitrogen, and in her calculation that there's got to be 33% more reduction just to keep your head above water and stay where you are. And this is just from that same paper showing it's both the Northeast and also the Midwest are sort of the epicenters of this. Okay, but there are other consequences of climate change and warming temperatures in our oceans. And that is that organisms, to stay in their thermal optimum, have to migrate towards the poles, right? Every organism, including humans, have their optimal temperatures, but the ones in the ocean that can't turn on the air conditioning will just follow that colder water and move towards the poles, and obviously move north in the northern hemisphere. So I want to talk about a marine organism that's well adapted to warm temperatures. That's Vibrio vulnificus. And what this is showing is the incidences. It's a bacteria, it's a pathogenic bacteria, and it's showing that can, I'll talk more about momentarily, can infect humans, but you can see the cases that occur in the very warm areas in this country. That's Florida and Texas, hundreds of cases per year. They track temperature perfectly, right? It's not an issue in colder months, becomes a big issue in warmer months. So the organism likes warm water, brackish waters. It must be in salt water, though, not in fresh water. It can infect open wounds, and the infections can be very, very serious. This is just CDC data of the outcomes when people get infected by this bacteria. Things progress very quickly. Now, typically the people who actually don't survive this are immune compromised or older, but still it's a very serious outcome. Typically it's when people have open wounds and go into water. And just to show, just last year with a hurricane that occurred following that and all the freshwater input from it, there were a lot of cases of Vibrio vulnificus. So this organism was only found in the Gulf of Mexico through the 20th century. So all the cases were down in the Gulf of Mexico. So I showed you Texas and Florida, that's where it's most common. What's happened through this century is that the cases have slowly crept up the East Coast. The most recent cases up until this paper was published, this is a 2023 paper, were in New Jersey. This same paper then did some modeling and said, well, based on CO2 emissions and year, when will the Vibrio vulnificus cases get into the Northeast? And so the answer was, well, it'll be around the year 2100 if we have high levels of CO2 emissions. Well, they were off by a little bit because actually the first cases were actually in 2023. And CDC, based on this, in New York, and based on this, the CDC issued this emergency preparedness and response document. Although I want to emphasize all the cases in New York, and I think the large majority of cases have nothing to do with shellfish. It's typically, again, people with open wounds going into water and then having those open wounds be infected. So that's, I think, very important. I think this headline got it wrong, and I think there's no evidence of this happening in shellfish in New York. But there is evidence of it of people getting infected. This was unfortunately, there were deaths. There were also people who got wounds infected and sick from that as well. So this organism is found throughout our waters. This is a survey we did immediately after the occurrence in 2023. We looked at some samples, found some hot spots. And since that time, and I'll just emphasize that, a high level is typically considered more than 1,000, so the orange and red. There was a study, for example, in Chesapeake Bay where that was like the peak that they found. You can see we had plenty of those, or several of those, and plenty of red hotspots as well. In the last 2 years, we've been tracking it further. And again, we've been finding it on the North Shore and on the South Shore, particularly in some of these. It likes brackish water, and so some of these, I'll call them salt ponds, have lower salinity and are more vulnerable. And again, we saw similar things, similar levels in 2025 as well. So it's in our coastal waters. And again, it's typically people just talk about it being, well, it's because the water's really warm in summer and brackish. And that certainly holds true. Here's all our data showing that, we don't see it at all below 17 degrees centigrade, and a significant inverse correlation between salinity of the water and the amounts of Vibrio. The unexpected outcome with regards to Vibrio in our waters is its response to different other environmental variables. So here's an experiment where we took water that we knew had decent levels of Vibrio. And again, I told you anything up in the thousands is a lot, so this was a lot. And then exposed it to either raw wastewater, it was filtered through a 0.2 micron filter, but that's it, or wastewater that had gone through a sand filter. And the outcome was something I'd never seen experimentally. I mean, bacteria grow fast, but you can see here in just 24 hours, going from tens of thousands to millions of these cells. So again, the wastewater was like supercharged these organisms. And in a similar experiment we did this year, thinking that it was what we call the organic matter in the wastewater, we did a similar experiment with a similar outcome. In that we added glucose, and that's just sugar, right? And just seeing, is it the organic matter? And well, yeah, it is a little bit, but the mix in the wastewater, it's more than just the organic matter, it's probably the nitrogen and phosphorus as well. We also I should say these were all added at equal amounts of carbon. So if it was just the carbon that they were growing off of, the bars would be the same size. But there were significantly higher levels in the wastewater, and this algal organic matter is simply just taking an algal bloom and lysing it, right? And so in that regard, it's both the wastewater directly or the wastewater promoting an algal bloom, lysing, and then leading to this supercharged growth of the Vibrio vulnificus. And just to show, we've done many experiments. I'm highlighting just some here in the South Shore. In the summer, nitrogen is always going to give you more algal biomass. And again, that more algal biomass is fuel for the Vibrio. So it's a complex story with Vibrio vulnificus, but we can say we have warmer temperatures, we have more rainfall, and certainly on-site wastewater is an issue as well. Okay, so I want to move towards, clearly there are some problems. What can we do to fix them? And I'm going to start on the septic issue. There are more than 400,000 septic systems that are draining into our drinking water source, our groundwater. How is that going to get fixed? Suffolk County does have a plan for getting that fixed. And they now, thanks to legislation that was passed by more than 70% last year, have a funding mechanism to address that. Their sub-watershed plan has prioritized where upgrades should happen. And again, I always point out to this map to show that this was done very, very carefully, right? The first thing they did in making this plan was saying, where are the public water supplies? Okay, let's make sure those areas that are getting wastewater into them that are our drinking water wells, let's protect those. And then secondly, you can see that pink is the highest priority. Those, if you just focus on that alone, those are the areas that should be upgraded first. And you can see, it's not like, let's just upgrade everything. In fact, look at the North Shore. There are plenty of places on the North Shore going to Long Island Sound. They said, not a high priority, right? But the South Shore is a whole different system, right? And the reason there's so much focus on the South Shore is because it's a very shallow water body. It doesn't have a lot of tidal exchange, has a long, what we call residence time. And so when that nitrogen gets into this system, it has a long time to lead to these water quality impairments. And to the converse, there's much more tidal flushing on the North Shore. It's a deeper water body, and therefore, nitrogen loading on the North Shore is of little consequence. And of course, the water quality outcomes match up with this as well. We're seeing the harmful algal blooms, for example, across the South Shore. We're not seeing them in the open waters of Long Island Sound. And in that same study, they modeled out how what might happen to those high levels of nitrogen that are the dangerous levels both for ecosystems and for human health. You can see that plan would lead to those areas vanishing. So there are several septic systems that have been approved by Suffolk County for install. I'll emphasize the huge amounts of money available for people to upgrade their septic systems at this point. You can read all the details here, but it adds up to, if you live in Suffolk County, there's $40,000 available to you to upgrade your septic system from the state, the county, and the county. And if you live in East Hampton or Southampton, it could be up to $65,000. And if you look at the Suffolk County website, that will cover full installation in many cases, including and backyard restoration. So, and I'll say, the state funds in particular are enormous right now. That makes up the bulk of the Suffolk County money. That is not necessarily permanent. So it's a good time to take advantage of that. I do direct the New York State Center for Clean Water Technology at the university, where we're harnessing science to engineer clean water to protect public health and the environment in New York and beyond. And I'll take a moment to talk about our nitrogen-reducing biofilters, or removing biofilters, that are made out of sand and wood chips, essentially, to create nitrification layer in the sand, and then in the wood chip layer, denitrification to remove the nitrogen. So a very simple system. Outside of that is PVC pipe and a single dosing pump that we literally get at Home Depot. These are the concentrations of nitrogen coming out of different systems. Then the green that have been approved in Suffolk County, ones that haven't been approved yet. The brown shows you if you do nothing. This is the average level of nitrogen coming out of septic systems. Our two systems have been the best performing in Suffolk County to date. We have two different varieties. Our box system is the one that is really doing well. That's shown here. In this case, the sand layer is separated from the wood chip box, but it's getting incredible performance and consistent performance. And beyond nitrogen, of course, you can imagine there's lots of other things in wastewater we don't want in our groundwater. These systems have been shown to remove very large amounts of emerging contaminants, so drugs, pharmaceuticals, personal care products, solvents. The removal efficiencies that we're seeing here are as high as or better than what you get from a sewage treatment plant. And these two systems are now approved in Suffolk County, and we have people who do install these. I should just say, of course, that we have no affiliation with this group. It would be XCAV who does the installations. Okay, and then we're also working on other things that can mitigate nitrogen loading. And one of those is thinking about wastewater as a resource, right? We spend a lot of time thinking about getting rid of wastewater, getting it far away as possible, getting it deep into our ground or far offshore, but can we think about wastewater as a resource? And one way to do that is via the process of fertigation. I think some people I was talking to earlier in the room are familiar with that, but the idea that wastewater maybe gets reused. And specifically, we wouldn't, I will say on the West Coast, where the people are in drought and water is extremely scarce, there are full cities where no water goes anywhere but reused. Nothing goes to ground. You don't waste a drop. Everything gets reused. So in this case, we're not talking about drinking the wastewater. That does happen on the West Coast. But just can it be used to fertilize lawns, for example? Rich in nitrogen, is there an opportunity there to collect that and spread that out to a golf course? And I'll say that actually this has been done, and that Frank Russo, who's my associate director at the center, designed the first ever system to reuse wastewater to water a golf course. Specifically, this is the Peconic, on the Peconic estuary. The Riverhead Sewage Treatment Plant discharges directly to surface waters. Right? So that's fresh water that was on land going out to sea. But instead, after being highly treated, so a lot of nitrogen removed, deactivating the viruses, deactivating the bacteria, it's now being used to actually water the Indian Island golf course. And something we're looking at closely now is actually at Stony Brook University. I don't know those of you who are familiar with the campus or not, but in the corner of the university, tucked in, is the sewage treatment plant that actually collects water not just from the university but from other neighboring regions and sends it off into Port Jeff Harbor, which has been a problem, frankly, over the years. We're looking into the possibility, as you can see, that sewage treatment plant is conveniently located next to all sorts of sports fields and open fields that get watered all the time. So it's the same opportunity. Another water reuse approach that we're looking at, and again, just emphasizing, this would take nitrogen, instead of it going out in surface waters, would have it be used on lawns. And then another thing we're looking at is, can we collect urine? And that might sound crazy, but interesting fact, 70% of the nitrogen in wastewater is in urine. And as it turns, nobody's talking about going to your home and giving you a special container. But as you may know, in the restaurant today, and in fact any public facility, in the men's rooms at least, there are urinals that collect urine. Now that usually just goes to wherever the wastewater is going, but it doesn't take much infrastructure to be collecting that. And that's done in other places. Certainly it'd be something you'd want to pasteurize and sterilize before use. But there are opportunities once you do that then to potentially use that as a liquid fertilizer or to capture the nitrogen phosphorus onto a solid and use it as a solid fertilizer. And just to show an example, this is an experiment we've done at the Center for Clean Water Technology, essentially showing the dose response that you can, how green do you want your grass? How much do you want it to grow? Well, you can see here, you can 4x your lawn production with the right dose of urine that's been sterilized. In fact, get an outcome better than commercial fertilizer. And you can see, maybe you can see in the lower corner here, the outcomes. So, and this is just one approach. Another approach, again, is to actually extract the nitrogen phosphorus out of the urine into a solid that then can be applied as well. Okay, now just to transition as I wrap up here, I just want to talk about seaweeds momentarily. As photosynthetic organisms, we know they take CO2 up, they take up nitrogen, they put out oxygen, and they actually can even fight harmful algal blooms. And don't need to review, but just, of course, we want less nitrogen and CO2 in our water. CO2 leads to low levels of pH and acidification that's harmful to bivalves. I showed you all the sites where we need more oxygen. So the kelp can help on all those fronts. Now, I won't go through the details here, but I'm going to point to some papers, two by my former research technician, Craig Young, who now works for the DEC, gainfully employed there. He's the lead author in these two papers that essentially he was able to show if you co-cultivate bivalves with seaweeds, kelp, or even ova, they grow better, particularly in the face of acidification. Right? So again, I'm not going to go through the details here, but I encourage you to look at these papers. My graduate student, Lane Silvers, and I published these papers that essentially showed that different seaweeds, including kelp, including ulva, have strong inhibitory effects on harmful algal blooms. And that if you co-deploy these seaweeds with bivalves, not only can you get rid of the harmful algal blooms, but you can reduce significantly the amount of toxin that gets into the bivalves as well. And then, so they're helping with harmful algal blooms, they're helping with acidification. And I want to point out one other outcome here. So here's an experiment, a lot of graphs to look at. But this top one here, A, essentially just showing by co-growing in this case it was mussels, with S. latissima, which is kelp, we can double the growth rate of the mussels. Now, based on our prior research, we assumed, oh, well, that's because the kelp must have fixed the pH, or fixed the oxygen, or got rid of a harmful algal bloom. Well, in this case, yes, it raised the pH, excuse me, the DO, yes, it raised the pH, both on average and at different times. But actually, the oxygen levels were perfectly fine here. And so the emerging research here is that these seaweeds are actually a food source for the bivalves. And as they're co-growing, there's actually both cells that are growing on the seaweeds and the cells of the seaweeds that are getting taken up by the bivalves that actually boost their growth. And at a biochemical level, bivalves need a certain complement of fatty acids that they cannot get strictly from filtering the water, right? Phytoplankton don't have them, but seaweeds do. And so seaweeds are actually sort of like a vitamin pill almost when they're co-grown with the bivalves. And this leads to something I call the halo effect, where the co-cultivation of seaweeds and shellfish have many positive outcomes for the bivalves with regards to harmful algal blooms, ocean acidification, and just additional nutrition. And I should say that this, I'm expecting will be published any day now. The last thing I'll just talk about is bivalve restoration work that we've done and the Shinnecock Bay Restoration Program. Again, you can get this publication. This is based on hard clam restoration in Shinnecock Bay. And the concept that we're all familiar with more bivalves, you can get clearer water, and that can benefit things like seagrasses, and can potentially then also be filtering out harmful algal blooms. So for the Shinnecock Bay Restoration Program, we planted 5 million clams, but I want to emphasize, and Bill Zeller's here, he played a huge role in supplying nearly all of the clams for this project. And the reason Bill was important is because we didn't rely on seed clams. Remember I told you, how long does a 10 millimeter seed clam live? It doesn't. You get like 0.1% survival. We use adults, and we've had, I won't say 100% survival, but in many of our places we planted them close to that for over a decade, right? Hard clams can live for decades. So this was a great, choosing the adults was a great investment because then specifically for clams also, oysters we're finding actually only, an old oyster is 5 years. Clams outlive them many-fold. And so we created what we called hard clam spawner sanctuaries, planting these hard clams very close together so that their reproduction would be successful. When we started, we recognized they're actually, hard clams are broadcast spawners. So when they reproduce, they just throw their gametes in the water. Successful fertilization means they have to meet each other somewhere. And if you don't have many of them out in the water, you're not going to get that. So we planted 50,000 clams in a half-acre plot so that when they did all spawn, they typically spawn on a temperature cue. Although if one clam knows another is spawning, they'll all get involved, so to speak. But when you said it, not me. But that leads to high levels of successful fertilization. And I'll also point out this was both a DEC and independently sponsored program. And this is just showing you where we planted these things. And if you know Shinnecock Bay at all, what you'll see is that we focused here on the western side of Shinnecock Bay. We specifically did that because we had what are known as hydrodynamic models that predict how are the currents moving. When clams spawn, their larvae are swimming around the water for about 2 to 3 weeks. So we wanted to make sure that wherever that spawning was happening, that they weren't being transported out to the Atlantic Ocean, but rather they'd be retained both within the western and eastern bay. So these maybe are a little bit reversed. So I'll just show that this is the filtration time. It took the clams to filter the water over time. That went down. So clams are filtering more and more water, or filtering it more quickly. Compared to when we started, the levels of algae in the water went down, phytoplankton, chlorophyll. The water clarity improved. And the clam populations have increased a lot. So firstly, just for adult clams, we had a 4-fold increase over time in this region, and also recognize that some of those larvae were actually transporting to the west as well. But the real action was in the juvenile clams, the small clams. And you can see that we separated into western and eastern side of the bay. Turns out, if you would blow these up, you'd see the same thing here, it's just a lot smaller. Most of them were being transported to the eastern part of the bay, and the survival was best in the eastern part of the bay. And there's really beautiful trends where you get years of really nice sets of the smallest clams. There's some mortality, but then they transition to being larger clams. So exactly what you'd expect. Again, so from a baseline to very high levels. And the most exciting thing was something we didn't plan on whatsoever, and that is that we did this to improve the filtration of the water, improve water quality. And with that, check the box. What we didn't expect is the huge economic multiplier this has become. The landings of hard clams in Shinnecock Bay are up 35-fold from before when we started, right? And you see it with your eyes. I've been working on Shinnecock Bay for the last 25 years as a scientist. Now when you go out, there are clam boats out there. There are people clamming. They're making a living on this. We've estimated that the landings, there's been millions of dollars into the local economy from this. And again, there's all sorts of economic multipliers you can put in that put it at tens of millions of dollars. So a happy and unexpected outcome. And with that, we've seen a great expansion in seagrasses as well, more than 6-fold. Okay, I'm done. I'm talking too long here. So wrapping up, I showed you that high levels of nitrogen, particularly from wastewater, is a threat to coastal ecosystems and human health. Climate change is accelerating, and when you couple that with the nitrogen, it has a compounding negative effect on ecosystems and economies. Upgrading septics is a primary mitigation strategy for improving water quality. And things like water reuse, using seaweeds to cultivate and bivalves as well can all work together in the meanwhile to also improve water quality. So with that, I'll thank you for your attention.